Once we have adhered to a host, we have basically three different ways that we can invade. Some pathogens, wherever they adhere, that's where they may set up shop. So it's not much of an invasion, but it's basically an invasion is the site that I'm going to start to proliferate and grow more of me so that I can become more numerous and pass on myself to another host so I can keep this cycle going. So some pathogens, wherever they adhere, that's where they stick. That is called extracellular invasion. Some of them may also stay at that same cell, but instead of staying on the outside of the cell, may go inside the cell. That would be an intracellular invasion. Or third, some may go deeper. So they may adhere to the host at a certain place and use it as a starting point to find another spot in the body in order to set up their shop. These are the three basic ways of invasion. I'm going to go into a little bit more detail on how they go deeper. So in order to go deeper, you need to be able to move. And this is in table 10.5. Marker's not being very cooperative right now. So table 10.5 are what are called different forms of invasins. Invade, invasin. Just like adhere, adhesins, invasion has invasins. So we've already talked about one. Different forms, ways you can get around. Those flagella. We talked about their function when we talked about the prokaryotic cell, and their function is to help move the cell. Matter of fact, in the picture above, you can see some that look like they have some peritrichous flagella, and you can see in the going deeper and passing between cells, they're using those flagella in order to move themselves along. Another way you can go deeper is by having certain enzymes. One of those types of enzymes are collagenases. Collagen is protein that is in the tissues of your cells. If you want to go deeper, you can break down that protein and move deeper into the tissue. Another one you can use are coagulases. So coagulases, these are enzymes that promote blood clotting. When we talk about blood clotting, we mean it congeals together. So if you have coagulases, you can actually make yourself a little shuttle. You can cause a bunch of blood cells to sort of make you a little spaceship, and then you, the pathogen, can sit inside, and now you can allow the blood circulatory system to sort of move you around and get you to where you need to go. On the other side, you have kinases. I want my black one. Kinases are actually going to break down blood clots. So one of the things that your body does to protect itself sometimes is to make little blood clots, because then that stops up the circulatory system. If you stop up the circulatory system, you can keep pathogens from moving along. But if you have a kinase, you can break down that blood clot that the body has put up as a roadblock, and then you can continue to pass on through. I'm not going to worry about neuraminases. These are ways that different pathogens can get into a particularly cartilage tissue. That's a pretty specific one. So if you know these 4 different types of invasins, you're good to go. Once you get to that site where you will set up shop and proliferate, in order to proliferate, you need to get nutrients. So the other half of this step is you need to make and get yourself nutrients. Just like you as a human, unless you get nutrients, you are not going to be able to sustain yourself, much less have enough energy in order to procreate and make more of you. Well, the same issue occurs for our pathogens. And the most limiting nutrient for pathogens is iron. So they have to really figure out how to get iron. You know, you can have all the glucose in the world and make all the ATP you want, but if you don't have the iron to then put other things together, it's just like any factory situation. Whatever you have the least of, that's the most you can make of that particular product. So you can have 500 extra parts of everything, but whatever is the limiting factor is the one that will slow you down. The way that pathogens have figured out how to get around this, in particular bacteria, is they have a little component called a siderophore. And a siderophore is a way for bacteria to obtain iron from their host. So it is a protein that can go out into the host system and it can snag an iron molecule and bring it back to the pathogen.